Today in this lecture, we will talk about the role of late distal tubule and cortical collecting tubule in formation of urine. And there are two different types of cells in late distal tubule and cortical collecting tubule, the principal cells and intercalated cells. We will focus our discussion on the importance of principal cells in this lecture, and then in the next lecture we will discuss intercalated cells. As we have discussed multiple times that urine formation begins with filtration in the Bowman's capsule. The filtrate then moves through the proximal tubule, loop of Henle, the different segments of the loop of Henle, the descending segment, the ascending segment, the thin and thick segments of the loop of Henle. Then it enters the early segment of the distal tubule, and finally late segment, a late distal tubule and cortical collecting tubule. Every part of the nephron has its own function in concentrating and diluting the urine, and especially conserving the total amount of fluid in the body, which is important for urine formation and maintenance of the different body fluids. Now coming towards the role of late distal tubule and cortical collecting tubule. Now both of these segments, the late distal tubule and cortical collecting tubule, their functions are more or less similar to the early distal tubule. That is, they will not reabsorb urea. They will allow the urea to move out of the body. They will allow the urea to move out. They will not reabsorb urea. Similarly, they will not reabsorb water. They will reabsorb sodium. They will reabsorb sodium, and some cells will secrete potassium and some cells will secrete hydrogen, but they will not reabsorb water. So water will also be allowed to move, and in this way they also act like diluting segment. Now how this part, how the late distal tubule, how the cortical collecting tubule, and how the principal cells present in these different parts help in reabsorption of sodium. Now the principal cells present in the late distal tubule and cortical collecting duct tubule has been shown here. This is a principal cell. On this side we have the tubule. The lumen of the tubule is present on this side, and this side is the basolateral membrane of the cell. So here we have the basolateral membrane, and here we have the tubule. Now the principal cells have two different channels. One is the sodium potassium pump, the other is sodium channel. The sodium potassium pump, it continuously throws out sodium out of the cell. It continuously throws out sodium out of the cell and brings in potassium into the cell. So it continuously increases the concentration of potassium in the cell, and it continuously decreases the concentration of sodium in the cell. When the concentration of sodium decreases in the cell, the sodium present in the tubule, the sodium present in the lumen, the sodium present here, it starts getting reabsorbed into the cells and start moving towards the peritubular capillaries here. But this movement is with the help of sodium channels. When sodium is moving in, it also helps in the secretion of potassium out into the tubule. So potassium is secreted into this tubule. Potassium is secreted here into the tubule while sodium is reabsorbed, and this process is done with the help of sodium channels. While initially the sodium channel is thrown out against the concentration with the help of sodium potassium pump. Similarly, potassium is thrown in against the concentration into the cell with this help of this pump. Both this sodium channel pump and the sodium channel has a clinical significance. Now aldosterone is a hormone which acts on this sodium potassium pump, and it enhances the activity of this sodium potassium pump. When the activity of aldosterone is blocked with the help of aldosterone antagonists like spironolactone and eplerenone, the aldosterone is not able to act on this sodium potassium pump. This pump is not able to function. It cannot bring in potassium. It cannot throw out sodium. So sodium is not reabsorbed. Potassium is not secreted. Hence, it leads to the diuresis because sodium goes out in the urine. It increases the amount of urine. Similarly, it prevents the secretion of potassium. So these drugs, these diuretics, they are known as potassium sparing diuretics. Now one category of potassium sparing diuretics, which includes spironolactone and eplerenone. They are aldosterone antagonists, and they act on this sodium potassium pump. The other category of potassium sparing diuretics include sodium channel blockers. They block this channel. They will block this channel, and when this channel is blocked, it again leads to no reabsorption of sodium from the tubule. So sodium will go out in urine and causing diuresis, and no secretion of potassium. So again conserving the potassium. So again it is included in the potassium sparing diuretics, and the examples of the sodium channel blockers are amiloride and triamterene. Now both these categories of drugs, the potassium sparing diuretics, have important clinical indications in different diseases. But their mechanism is simply in the late distal tubule and cortical collecting tubule, and the function is to block the sodium potassium pump and sodium channel. Now to summarize, the late distal The late distal tubule and cortical collecting tubule will not allow reabsorption of urea, it will not allow reabsorption of water, and it will allow the reabsorption of sodium. But there are two kinds of cells. The first category is the principal cells, the second is the intercalated cells. Principal cells have been discussed in detail. They help in the reabsorption of sodium and secretion of potassium. Intercalated cells will reabsorb potassium and will secrete hydrogen. We will discuss them in detail in the next lecture. And again, the different channels, pumps acting in the principal cells can be blocked with the help of aldosterone antagonist and sodium channel blockers. And that's the clinical importance of principal cells in the late distal tubule and cortical collecting tubule. Thanks a lot for watching the video.